What is the purpose of an enzyme? The purpose of an enzyme under most conditions is to actually help reactions, chemical reactions, go more effectively. In particular, what we are focusing on is something known as the energy of activation. What does this actually mean? It focuses and targets the energy that is needed to break bonds and to form new bonds. So the energy that is involved in either one of those processes is referred to as the energy of activation. Think of it like this. If we have a ball or a molecule that you need to move uphill to a higher energy level, what we need to do is find a way to make that reaction happen, but to not be extremely costly to your system. So what we have to think about is, how can we make reactions go faster? Many of you might be thinking about heat. You can heat up a system and the reaction will go faster. What happens if we heat up the inside of you? You die, or your cells die. So we have to think about limiting heat. So what are our other options? We've got a couple, but what we want to focus on today are enzymes. Enzymes' primary role is to lower activation energy or to stabilize some sort of transition state. In my picture here, I'm showing you how we can break down lactose with lactase. This is a reaction we have now talked about several times. And by using lactase, that enzyme, breaking down this sugar, lactose, is easier because of that enzyme. So it's lowered the amount of energy your system has to put in order to break that bond in lactose and make simpler sugars that can be used in cell respiration. Without the enzyme, it takes a huge amount of energy to come in and actually break that bond. What we want to look at is, what's the energy savings? What can we do because we used enzymes here that have saved us a lot of energy, and now we can do more work? Enzymes we usually look at as being proteins, as we've mentioned. They don't have to be. We can also look at RNA structures that we'll talk about later as acting as proteins, as enzymes in particular. So in DNA replication, we'll look at something known as tRNA and rRNA. Both of these are actually functioning like enzymes. So it doesn't have to be protein, but it most often is. The idea here is that we've got some reaction that is occurring. The amount of energy is on your Y, and how fast that reaction is going is on the X. So we start out with reactants of some sort, and without the enzyme, the black line, you see the amount of energy required is up here. With the enzyme, the red line, you see the amount of energy that we now need is down here. The difference between the two, this space here, is actually A minus B, gives us the energy savings in this reaction. Now we still end up at the same point. This is important to remember. We can't lose energy or save energy and then come out with a different product. That's not going to work for us. We still need to end up at the same place. Chymotrypsin is a common example of an enzyme that's going to actually help break down and process actually tryptophan in your pancreas. So when we break down tryptophan, so the chymotrypsin is going to work on tryptophan and break it into monomers. The whole idea, remember, of breaking down polymers is to eventually break them down into monomers. But the idea here is this active site is where we're actually going to attach the tryptophan. So the tryptophan is actually this chain that's going through. The active site stresses the bonds right here, and when it does, water easily moves in and helps to break that bond, and all of a sudden you have two pieces. And we keep this going and going and going. And the idea is we keep cutting this chain until we get down to having individual monomers that we can then use for other things. How fast this happens doesn't have anything to do with whether or not we're putting in or taking out energy. It has everything to do with the interaction of the enzyme with the substrate, or what it's attaching to. Okay, so what we're really looking for is enzymes to act as catalysts. We want the enzymes to make these reactions go faster. In this process, we're allowing other things to occur because these reactions went faster. So with enzymes, shape is critical. So we've talked 3D shape before. Critical for what we call enzyme specificity. So that is the matching of an enzyme to its substrate, or what it's going to attach to. So that specificity, how strong is that connection? Does enzyme A always pair with substrate B, or are there other options? So what we have to look at is something known as that active site that I was mentioning to you before, that's part of the enzyme and helps to attach that substrate to where it belongs. Let me show you. This active site in this case is specific for sucrose because the enzyme is sucrase. We've talked about some of these matching ones before. So sucrose fits into that active site. The enzyme, when matched with its substrate, actually is going to stress the bond of that substrate. When it does, water can come in, help to break this apart. We suddenly have glucose and fructose, two simple sugars that can go straight into cell respiration. This is a critical enzyme for us in helping to break down fruit sugars and so on in our systems and make the sugars more available. Here's an example of hexokinase. This is the large enzyme responsible for helping us to break down glucose right here in the middle. So you see how large the enzyme has to be in this case to help us break down glucose. In many cases, these enzymes are so large that they might have more than one active site. We see this quite a bit, and we're going to talk about this as far as enzyme regulation in a little while.